So next, I'd like to introduce Matthias Heikenwalder from from the German Cancer Research Center in Heidelberg. Matthias has done really great work on both HCC and cholangiocarcinoma, the role of inflammation, interplay with the immune system, and cancer initiation and progression. And looking forward to your talk, Matthias. Thank you.
Thanks for the kind introduction and also the invitation. Really enjoyed the meeting so far. So really happy to be here and learn, actually. It's a very special meeting also to be able to talk to patients and to see actually how this affects individual people as well. So I'd like actually to put into focus something that has not been talked about in that detail, and that is actually the microenvironment. All these diseases, or in this particular case, cholangiocarcinoma, can occur. And although the fact that we have heard a lot about mutations and how important they might be for cancer development, I should point out that mutations that induce proliferation of tumor cells, prolong survival of tumor cells, we should not forget that these drivers actually also affect how these tumor cells interact with the environment and how the environment around these tumor cells interacts with the latter as well. So to give you maybe a short summary, what we already know and what has been also discussed, and where it's actually not pretty clear for a lot of etiologies how they affect CC development. From hepatocellular carcinomas, it's pretty clear that there are several etiologies that can drive different subsets and subtypes of HCC, and it is also well known and has been discussed there are particular etiologies that can also drive cholangiocarcinoma. I should point out that we should not forget that all these diseases, in most of the cases, develop on the basis of a chronic necroinflammation, or at least a chronic damage and stress that these cells have to suffer from. We usually what we do is we try to mimic the human pathology by first looking actually at human specimens, analyzing the inflammatory signature or particular things that we see to be different. And what came to our surprise, or what was really interesting to see, is that as has been discussed today, CC and HCC can even coexist. So as if there is a battle between those two primary liver malignancies. So we were wondering whether there is something overt that we could find that is different between hepatocellular carcinoma and cholangiocarcinoma. And what we found to be striking, although I don't want to say that it is mutually exclusive, is the fact that when we looked at cholangiocarcinoma or hepatocellular carcinoma, that we found an increasing incidence of reactive oxygen species, ROS, in CC when compared to HCC, which somehow indicated that maybe on the basis of a very high ROS, cholangiocarcinoma have a higher propensity to develop. So based on that hypothesis, we actually took the next step and asked, is actually this reproducible in mouse models in which CC can be or have been developed? And as nicely introduced by my previous speaker, there are several models that have been using the hydrodynamic tail vein injection model or a CRISPR-Cas model where actually several different mutations were introduced. And in all of those cases, we could to some degree confirm that we see these high ROS levels within the lesions and nearby the lesions in those livers. What was striking to see that, for example, in models that at the same time also developed hepatocellular carcinoma, the high levels of ROS were only to be found in the cholangiocarcinomas. So based on that, and keeping in mind that we wanted to generate a mouse model that might be wrong but maybe useful, we thought about generating a mouse model that has high ROS levels in liver cells. And HSP60, it's a heat shock protein, has actually been identified as a gatekeeper for keeping ROS levels low in hepatic cells. So we set out in collaboration with Dear Carla to generate a mouse model in which we can specifically deplete genetically by Kree-Lox recombination HSP60 in hepatocytes or liver cells. In comparison to the previous speaker, a very long-lasting experiment. It takes 1.5 years until you have actually a colony you can work with. So this is maybe details we don't have to go into that much, but what I would like to point out, when you then perform molecular biology controls to investigate whether indeed you could knock out that gene specifically in cells of the liver, we have been successful, as you see that there was a reduction, a massive reduction, mRNA transcription or protein expression. And even if you look at immunohistochemistry, we could see that in contrast to the wild-type situation in these HSP60 delta LPC mice for liver parenchymal cells, we could actually massively decrease expression of HSP60. So there was an efficient depletion in those mice. So the question was now, what's going on in these mice, and could they be a model to work on some hypothesis maybe interesting for CC development? Turns out that actually at the very beginning this was a very devastating result, because actually the mice died, succumbed to death after 10 weeks, and obviously the PhD student working on the project was not that happy with. But it turned out, as you will see, it was a very good model to actually start some hypothesis which would help us then to understand mechanisms of CC development and the microenvironment. So these mice are, as you see, smaller in size. They have much less weight. They succumb to death between 8 and 10 weeks. But what was already very striking is that they have massive fibrosis after 8 weeks, which we usually in these mouse strains seldomly see. They suffered from jaundice, cholestasis, so the serum becomes yellowish, and had a very interesting liver to body weight ratio. In addition, what was striking, and what you can partially also see in human specimens, that in addition to our aim to induce high ROS levels in these animals, we found signs of mitophagy. This is just an indication that mitochondria are engulfed by additional membranes and, so to speak, eating up themselves, and that's what we call mitophagy. Histopathological feature. So in addition to that, these mice really suffered from a lot of damage. So, and damage you can see by hematoxylin and eosin staining. For example, here this is massive necrosis. Interestingly, this death of cells was not induced by any classical apoptotic cell death. It turns out that most likely that this is another cell death, which is called necroptosis. We had a lot of fibrosis ongoing, what you would see also in the human specimens. And what was also striking is that the hepatocyte function was massively reduced, as you can see here, beautifully as indicated by cholesterol and triglyceride levels. So now the question was, and that's the interesting thing about this model to start with, that when we looked at histology in this very short time frame, we had this approximately 10 weeks, that it turned out that the cells that proliferated the most were actually not hepatocytes, it was cholangiocytes. And when we looked over time, it turned out that these mice developed, as you will see, perineoplastic lesions, so-called bins, that you have actually heard about this morning before, and this is actually how they look like, and this is how they can be quantified over time. So these biliary intraepithelial neoplasias that we have seen in the mice are actually reminiscent of those that you have seen this morning with different stages. Very difficult to define stages that are congruent to the human situation, but it turned out that indeed there are these bins to be found. So, of course, you could say, well, this is hematoxin eosin, the guy should show us that this is really the case on an immunohistological level, and that is really true. There are lineage markers. We can actually identify those lesions as well as particular tumor markers, CD90.1, CD44V6 for actually, let's call it perineoplastic cells, and for example, these cells were also positive for nucleus MYC, as were some hepatocytes that were in vicinity to those lesions. So really looked like what you could call a perineoplastic lesion. So then what you do to really show, first of all, that genetically these already only 10-week-old lesions are reminiscent of some genetic lesions that you see in humans. You have to laser dissect those structures, and this is what we did by laser dissection microscopy. And then we performed with a low amount of samples area comparative genomic hybridization and then also exome sequencing to find out, do we actually have mutations in these lesions? And yes, this is the case. And when you then do a synteny analysis, so that means you compare the genomic data that you get from your mouse model to TCGA databases from humans, turned out that there were a lot of chromosomal aberrations, deletions or amplifications that were reminiscent of what you would see in a human CC situation. so to speak, the alterations to some degree reflect the human situation. So then, of course, what we did in addition to exome sequencing, we looked at, okay, how does this actually translate not only in protein, but also transcriptomics, so in expression, mRNA expression levels. And it turned out that in an unbiased screen, the highest candidate was CMYC, but there are several other candidates you might know of, NRAS, KRAS, even FGF receptor was highly upregulated. So that was kind of interesting that when it comes to a transcriptional profile, we have seen that it to some degree mimics something that you would expect in a human CC as well. And of course, so to speak, the proof of the pudding is in the eating. So what you have to show is, although our mice did die after 10 weeks, that this is a real malignant tumor, and malignant tumors should be and have to be transplanted. So the transplantation is usually performed in immunosuppressive mouse models. In this particular case, we use skate beige mice, where you mince the tissue of these livers that have these pre-neoplastic lesions, and you transplant them into a recipient mouse. And then you wait, so to speak, the additional time which you would like to have investigated in a mouse that has already died, and you have a look whether within 6, 8, 10, 12, or 14 weeks, these structures really develop into malignant tumors. And that was really the case, and these tumors, as it turned out after talking to pathologists, are reminiscent of what you would see in an ICC, but of course, obviously, since they have been injected subcutaneously, have developed in an environment that is not reflecting the one that you would see in the liver as such. So by having now such a mouse model system that, might sound awkward to you because, this is a mouse that dies and we take out the tissue and we transplant it, we have thought maybe that model, because it is so short, could give us some hunch on how actually these preneoplastic lesions, this tumor initiation, is actually ongoing on a mechanistic level in that environment of massive damage, high ROS levels, and as you will see, high Kupffer cell activation. But before that, we really needed to know on a mechanistic level what actually is going on these preneoplastic lesions in these livers in terms of signaling, in terms of ROS activation, what pathways are turned on. So I'll rush through this because that might be not too interesting for you, but what is striking to say is that we have seen massive mitochondrial dysfunction in these lesions and in these livers as indicated by specific candidates for mitochondrial unfolded protein response. And one of those, or these genes that should be pointed out and noted on is CHOP and GAT34, for example. We then took a next step and we asked the question now, where does actually this ROS level occur? Since, as we have found out, not all cells in the liver were knocked out. And it turned out what was very striking for us to see, and just look at this is a HSP60 staining. So here we have a region that is full of hepatocytes and cholangiocytes that is positive for HSP60, and then you have a region that is completely knocked out. So maybe even mimicking a situation in human tissue where you have loss of expression in a particular region and you have high expression or normal expression in another region. And it turned out that in the region where actually HSP60 is deficient, this is exactly the region actually where you have high ROS levels. This is exactly that region here. However, where you have wild-type HSP60 expression, apparently turns out that ROS levels are very low. So indicative that in those regions where you have low HSP60 expression, you have high ROS, and as it turns out, the proliferative capacity of the cells is very much increased in the wild type, but to some degree also in the knockout regions. Now, turns out that indeed, as we have expected, oxidative stress and the components of oxidative stress that are listed here are heavily upregulated in those livers and regions where we have the high ROS levels. So HSP60 deficiency leads to oxidative stress. This is what we would have expected. And as a consequence of this, and it has been discussed in some of the talks today, we set out with the hypothesis, why not actually blocking oxidative stress by giving mice a diet with an antioxidant. And of course, you could use vitamin C, you could use vitamin E, but we have been using a very strong antioxidant drug. It's butylated hydroxynanisole, it's BHA, and that has been used in some other cancer models where it has been identified that antioxidation could be beneficial. or blocking oxidation could be beneficial, and we've done this. We've fed the mice with the diet, and what you nicely see now is that mice that are fed the diet at the very beginning also lose weight. So this is a very macroscopic analysis, not molecular biology at that stage. But what was striking, that now they gain weight over time, and they do not succumb to death with that high incidence, as you see here in the Kaplan-Meier curve, with 100% incidence after, for example, 8 weeks. So we could rescue the mice in terms of survival. And moreover, what we could do is we could revert or prevent the phenotype from happening, which was this high fibrotic phenotype. And as we can show also, we could prevent the development of these lesions and the necrosis, which you see here. So this is the massive necrosis that we usually see in our knockout mice, and this is now small necrotic areas that we see in the treated mice. And most importantly, also the serum markers indicative of the damage that you would usually expect in such a mouse model with such a phenotype was heavily reduced. So hepatocyte cell death was significantly reduced upon BHA treatment. And so, most interestingly, was the aberrant proliferation of these preneoplastic lesions. And that was most or best indicated again by CD44 V6 expression, by CD90.1 expression, as well as by proliferation, and was then also quantified. Interestingly, I will show you this about the link to IDH1, which might be interesting for you, is the fact that in our mouse model, in our knockout mouse model, you see a high expression of CK19 and a very low expression of HNF4 alpha, as if this is down-regulated or even prevented from being expressed. Now, when we do the BHA diet, actually the things completely change, and you again have a high HNF4 alpha expression when compared to the normal knockout, whereas in contrast, the CK19 expression is going down. And I think this is very important why these mice actually survive, because the hepatocytes apparently keep regenerating the liver again, and thereby, so to speak, the bias towards cholangiocyte proliferation is gone, and there is no development of these preneoplastic lesions. So showing that changes of the liver niche can be induced by blocking ROS, we took a next step and really wanted to know now what's really the environment, the cellular environment, the immune cells that actually are within this pathology. And we investigated actually genes from which we knew that they either drive hepatocyte or cholangiocyte proliferation. And the genes are well characterized. We have not done this ourselves. There were many groups that have done this. And so we performed a very easy experiment, a time course. So we took mice at different time points within these first 10 weeks and simply investigated what are the genes or transcripts of many transcripts we looked at that are strongest expressed in those knockout mice. And it turned out, interestingly, that TNF was one of the first and highest transcripts to be expressed in our knockout mice, giving somehow us a hint that these high TNF levels could have something to do for this transdifferentiation towards the cholangiocyte lineage. So we then first asked the question, okay, if TNF is an important player, then actually the BHA treatment that I've shown you before should actually dampen TNF. And that was really the case. You see here that the TNF on mRNA and protein level was reduced upon BHA treatment. And we then asked the question, okay, if TNF is expressed at high levels and might be linked to pathology, what are the cells that make TNF? And it turned out that actually the cells that make the high TNF levels are Kupffer cells or myeloid cells of the liver. Let's put it that way to be absolutely correct, because we cannot exclude that in addition to Kupffer cells, there are other cells that make high levels of TNF. And what was also very striking is that they make the TNF in a very focal manner. And this is just the comparison to the BHA diet, where in addition to actually getting rid of the TNF, we could also obviously get rid of the activated Kupffer cells and myeloid cells, which in our knockout mice are obviously still present. Okay, so if that is the case, and if there are cells, and apparently maybe really TNF might be functionally linked to the pathology, TNF is a secreted molecule, it's a cytokine, which has to be taken up by a recipient cell. and the recipient cell has to express the receptor. Now, there are various receptors that can take up TNF. It's TNF receptor 1 or TNF receptor 2. And to make a long story short, what we found out is that TNF receptor 1, against actually our belief, is not expressed to high degree on hepatocytes. It's actually expressed on CK, on cytokeratin positive cells, as you can see here in a wild-type liver even. And what was very striking, when you now then looked at these lesions, these preneoplastic lesions, they were highly positive for TNF receptor 1 and double positive in this particular case for CK19, and that could be also corroborated by in situ hybridization. So that indicated that most likely the cell that takes up this cytokine, which is produced by an innate cell, the Kupffer cells or some myeloid cells, is maybe a cholangiocyte. So, and that would make sense that then actually you really have maybe a functional link to this preneoplastic lesion development. And we then did some double in situs to show that the F480 make TNF, not so important. But then was really the most important experiment, and now come some experiments where you can really use this mouse model now to do functional analysis. And the first experiment we did is a very dirty experiment, I have to say. You can inject clodronate. It's a substance that actually depletes all antigen-presenting cells. They take up the clodronate in lipids, and antigen-presenting cells, they like to eat lipids, and they take the lipid up and then they die. And what actually happens is that very nicely, but in a dirty manner, you can get rid of Kupffer cells in a liver. So you can investigate and ask the question, are Kupffer cells important for the development of my disease, because of this particular disease? Because if you don't have any Kupffer cells, and if the Kupffer cells are the cells that drive disease, the disease should be gone. And that is really what happened. By just depleting Kupffer cells in this very dirty manner, we could actually not fully, but strongly abrogate these preneoplastic lesions, which you see here, and that was also, again, nicely quantified. So that actually somehow indicated that there are innate immune cells that secrete a cytokine, most likely it's TNF from our expression studies, that might drive this preneoplastic lesion development towards a cholangiocellular proliferation, let's call it like this. And if this TNF is really taken up by cholangiocytes or cholangiocyte-like cells that express TNF receptor 1, then genetic deletion of TNF receptor 1 should do the same thing. It should actually block the development of preneoplastic lesions, but since this is extremely downstream of the whole process, it should not affect Kupffer cell proliferation, and it should not affect high ROS levels, because the high ROS levels in hierarchy are still present, right? TNF receptor 1 is downstream. And that's what we did, and this is really also what came out. TNF receptor 1 deletion fully abrogated the phenotype, and the interesting thing, it was even in a expression level dependency. So if you have a hemizygote TNF receptor 1 mouse, so one allele is knockout, one allele is wild type, you get a phenotype in terms of Kaplan-Meier that is somewhere in the middle. If you knock out both alleles, you get a full rescue when compared to the knockout mouse that we had. So TNF receptor 1 expression really is important, and that was then also not only corroborated on macroscopic level, but also on histology, that you get actually a reduction in these preneoplastic lesions, as you see here. This is just a control for the lack of TNF receptor 1 expression. So proliferation is gone. This is an A6 oval cell marker. You've heard about this today for these cells that might actually contribute to the preneoplastic lesions. But most importantly and strikingly, as I've told you, since TNF receptor 1 is downstream, ROS levels were still high and Kupffer cells were still activated. So everything that we could do to reduce the proliferation of these cholangiocytes and to reduce these lesions that at the end then might form cancer were still developing or were still blocked in the context of high ROS levels and high Kupffer cell activation. And again, similarly to what I've shown you before, we could, so to speak, revert the HNF4 alpha expression level, and this is what you see here. So this is the HNF4 alpha expression level in the knockout. It's very low. It's actually going down over time. Now, when I knock out TNF receptor 1, but still have the context of ROS and activation of Kupffer cells, actually HNF4 alpha expression is going up, showing you actually that hepatocytes express the transcription factor that gives them the possibility to differentiate. So TNF receptor 1 expression is very important, and we could also show, I'll not go into detail, that indeed it is the cholangiocytes by genetic and other tools. So the question, of course, was what's now happening and what's ongoing within the cholangiocytes? So what is the TNF receptor 1 that has taken up TNF? What is it doing inside of a cholangiocyte that drives then this proliferation? And I maybe was very narrow-minded, but I thought that actually it must be REL-A translocation, because TNF and the cognate and most well-known signaling pathway induced by TNF receptor 1 is the canonical NF-kappa B signaling pathway, which goes through the translocation of a transcription factor, which is called REL-A. And that means that if that is the case, you can actually follow this molecule into the nucleus of a cell, and you could stain it within the nucleus of a cell. But it turned out that I was wrong, and the student was right, and that actually it is not a translocation of REL-A into the nucleus. This is here now hepatoblast cultures, but we did the same in vivo. But that actually it is the translocation of an activation of a pathway that is called ChainK, C-Jun ChainK signaling. And we can stain phospho-ChainK as well as phospho-C-Jun in the nuclei of these lesions, as you see here and as you do see here. So apparently what TNF is doing, it's turning on through TNF receptor 1 on cholangiocytes, a particular pathway within the cholangiocytes, which we can show in vitro. If you block this pathway, actually the cholangiocyte cell lines, they die within 48 to 78 hours, but I don't show you the data here. So this is a pivotal signaling. These cholangiocytes, these aberrant cholangiocytes need to stay alive. But to show this in vivo, I'll show you two experiments and then I'm almost done. I show you an experiment with an inhibitor. So now I take a ChainK inhibitor. It's an inhibitor that I can inject into the mouse, yeah? And I can ask the question, would the injection of this inhibitor suppress the phenotype or the development of this preneoplastic lesion? And that is really the case. When we do this from day 36 on to day 49, so this is for approximately 2 weeks, I can massively reduce the incidence and the severity of these lesions. I can massively reduce the proliferation of these lesions as well. Indicative of that, ChainK signaling might be really important. And that just shows you that we can even rescue the survival of the mice. But of course the question was, is this now a phenotype in our mouse model, or does this actually, can this be translated and phenocopied in all other mouse models of cholangiocellular carcinoma or cholangiocarcinoma? That was really the case. Here are again the 4 most used mouse models for this particular phenotype, and I'll show you in a second also that this holds true in human cohorts. So whenever we have the lesions, we have nuclear translocation of phospho-ChainK at those lesions, and in vicinity to those lesions we have myeloid cells, FRAD positive cells that make TNF. Turns out in the human situation it's not only myeloid cells, it's also fibroblasts that can actually make high levels of TNF. But again, I think the most important experiment, and coming back to the experiments that Scott has shown you just beforehand, would really be, if that holds true, you should be able to use a double knockout mouse for ChainK signaling, which we call ChainK1-2. You knock it out specifically in liver cells, so you use an albumin promoter, and then by hydrodynamic tail vein injection, you induce extremely aggressive tumors or cholangiocarcinomas, either by using p53 KRAS, as was introduced in the talk before, or AKT-Notch, which is a bit different in terms of differentiation and pathology. And what was really striking to see, that taking away ChainK1 and 2 from liver cells actually fully abrogates the malignancy of these tumor entity, and in the case of AKT-Notch, even the mice didn't succumb to death. In the case of p53 KRAS, approximately 20% of the mice did die, and when we then opened up the mice and analyzed them histologically and macroscopically, it turned out that the 100% incidence of tumorigenesis that was seen in the wild-type controls was reduced by 50% in the ChainK double knockouts. Similarly, when we looked at the p53 KRAS, it was approximately also 50%. So genetic deletion reduced CC incidence, tumor load, and prolonged survival time for these mice. So finally, I would like to come to the human data. So of course, then the question is now, is the phospho-ChainK or the ChainK signaling also strongly activated within human tissue specimens compared HCC to ICC. So again, we looked at approximately 200 patients for each primary liver cancer subtype and looked at TNF levels, and we looked at phospho-JNK levels, and it was really striking to see again that indeed similar to what we've seen with ROS, that, you know, I'm not saying that it's completely absent, I'm just saying that the incidence of highly positive primary liver cancers that are HCC is very low when compared to CCC, when we looked at approximately 200 patients. So to sum this up, what we actually could show is that this TNF that is expressed by Kupffer cells or myeloid cells actually provides important cues to liver cell plasticity. This is a mechanism of TNF that was so far not recognized, that under particular stress situations in a particular environment, TNF does not only execute cognate functions, it might also execute functions that go towards plasticity and transdifferentiation of hepatoblasts more towards the cholangiocytic lineage, and thereby driving proliferation of cholangiocytes and then maybe preneoplastic lesions. And further on, we believe that that particular axis, the ROS TNF JNK axis, might actually provide a novel target for ICC, but that of course has to be tested, and we're doing a lot of experiments as a consequence of this in mouse models, but also checking a lot of human specimens and looking for mutations that go together with what we see when it comes to phospho-JNK activation. So at the end, I'd like to thank all the people involved. In particular, just 10 seconds, this study was mainly triggered and performed by Datien Huan, and we have been doing this together with Dirk Haller and Achim Weber, who really helped us from the point of view of a pathologist. So thank you very much for your attention.